Question 2 it is. This one is giving you an elaborate outline of what it is that you're actually getting to have. Let's call it cell organelle soup. The instruction here is literally you're taking animal cells, you're cutting them open and in an isotonic buffer, bursting those cells open, breaking them open, then gradually centrifuging out the individual organelles that you could have inside an animal cell. So for all you know, maybe A could be mitochondria, B could be Golgi body, C could be endoplasmic reticulum stuff, and D could be maybe a few ribosomes or some lysosomes, and that would be the rest and leftover cell organelle, sorry, the cytoplasm stuff. So that could kind of be what you're getting, but you don't quite know. You just know it's the parts, the fraction of an animal cell. Which one's which one, you don't know. It might be that my order was completely messy because the lipid-based ones might be lighter and therefore not suspend as quickly. But literally you have got a cell organelle soup, literally cut open cells, and the different parts of it are in the different samples A, B, C, and D. It then gives you that beautiful table of the different samples that were centrifuged out. So again, you don't know, one somewhere between A, B, C, D, and E could be endoplasmic reticulum, another one could be ribosomes, another one could be lysosomes, and so on, so lots and lots of different cell fractions. Now what does it tell you? Each sample was then incubated with glucose and pyruvate. Okay, so you've got here, these samples are incubated with glucose, these samples are incubated with pyruvate. Where's the difference? Where would you get what working? The key links that you need to get in your heads already when you're at that point. Hold on a second. What was pyruvate? Pyruvate was the end of glycolysis, and that substance will then go into the mitochondria to feed the mitochondria and do the chemical reactions there, whereas glucose is literally the starting point for your anaerobic respiration in the cytoplasm, if that's what you wanted to crack on with. So this is literally what should go through your head at that point already. And what are the different products that were forming? Carbon dioxide, lactate, carbon dioxide, lactate. So this would be products of aerobic respiration, products of anaerobic respiration, aerobic, anaerobic. So this is just to understand the table. The first point has to always be understand what information you're being given. Based on these results, identify the cell components. So what is A, B, C, D, E? What cell components are there which are present in samples B and E? Explained how you've reached those conclusions. B is sticking out like a sore thumb because of this yes here. So let's have a look. I incubated with pyruvate, and it produces carbon dioxide if it gets pyruvate. What cell organelle will be doing aerobic respiration if you've got pyruvate there? Oh my goodness, B has to be your mitochondria based on this information. So because we obviously seem to have link reaction Krebs taking place, otherwise we wouldn't have carbon dioxide being produced at that stage. Okay, then what is E? How is E sticking out like a sore thumb? Let's have a look. We've got here that yes, that kind of says, hold on a second, there's something different to all the others. They've all got no. So whatever substance E is, whatever organelle or cell component E is, it produces lactate if given glucose. Where does anaerobic respiration take place? So you need to kind of put two and two together. Anaerobic respiration of glucose is taking place in the cytoplasm. So E has to be cytoplasm because that is where glucose is the substrate for the actual reaction. Just putting the mark scheme here that you can have a look at what's going on. As per the other one, just pause the screen or take a screenshot really quickly that you've got the mark scheme if you want to look at it slower. It's all uploaded for you anyway. Okay. Moving on to B. Cyanide inhibits, cyanide, for what you've learned about, that should already click in, that is a respiratory inhibitor. Inhibits an enzyme, cytochrome oxidase, and prevents the regeneration of NAD. Where is NAD regenerated? So it was reduced and then it's being regenerated. That happens at the electron transport chain in the mitochondria. So, explain why cyanide could be used to confirm which sample contained resuspended mitochondria. So hold on a second. The electron transport chain would be stopped. The electron transport chain is in mitochondria. So if you're adding cyanide on into sample B, you would no longer get carbon dioxide produced because NAD would no longer be regenerated. NAD no longer can go back to the previous stages, cannot go back to the Krebs cycle, can therefore not pick up any more hydrogen and electrons from there, and therefore the whole producing of carbon dioxide, the producing of carbon dioxide that you've seen here, would no longer happen. So let's look at the mark scheme to go with that. So cyanide would only affect mitochondria as cytochromatic oxidase is part of the electron transport chain. Therefore, we haven't got any more NAD that's been regenerated to be going around and picking up hydrogen as a hydrogen acceptor from the Krebs cycle. Electron acceptor is just as fine there. And therefore, no carbon dioxide would be produced. So all 3 marks for the whole thing. And last but not least here, and this is literally, look at it, if it says 2 marks, you have to have 2 parts in it. Explain where the preparation of cell fraction was carried out using an isotonic solution rather than pure water. That means explain what isotonic is and explain what would have happened if you had pure water. So step 1, isotonic, that would be a water potential equal. On both sides you've got the same amounts of water versus salt or water versus sugar. So you've got an isotonic, not hypertonic, not hypotonic. Therefore, you wouldn't have substances moving in and out, which means that your cell organelles that you are extracting by that method do not burst or lyse, go into lysis, because actually in pure water, if you had pure water that has got a 0 water potential, which means that what's inside your cell organelles is most likely more water potential negative, the water would move from the pure water into the organelles, and the organelles would actually go and explode, and you say your mitochondria might end up mitochondria debris and not actually the actual fully working functioning mitochondria. So mark scheme for you here. So the first part to explain what is meant by isotonic, and the second part to go into what actually would happen if you had pure water.